Hey
Yeah.
guys, welcome back to Alex Invest. We're here with Mountain Valley MD's CEO Dennis Hancock, and basically Mountain Valley is a company that takes existing drugs on the market and basically makes them better, makes them more soluble in some cases, and other things like that. So yeah, you've had some new developments here as well recently, Dennis. So maybe we'll start with the selamectin sort of stuff there and kind of what's going on there. And it's interesting because it's never been used in humans before, right? So this would be the first time with treating TB, or
Yeah, so great to be on your show. Thanks for having me. Yeah, lots of moving parts inside Mountain Valley for
yeah.
sure. Selamectin, we'll jump on it quickly, but I think it might require a bit, broader context before going that deep. But selamectin, essentially, we have a solubilization patent that we've applied
Yeah.
to this macro-cyclic lactone class. And so ivermectin is a very popular one that people have heard of.
Hmm.
And selamectin, of course, is it's a similar drug, a antiparasitic class of drug. What's interesting about tuberculosis is, of course, it's the number one sort of infliction, if you will, in this space globally. 1.4 billion was sort of the last thing I had seen, and some significant death counts each year. So we're doing some interesting work now with selamectin. We had on our whiteboard for a while to go and pursue that solubilization technique first. And then I'm not sure if you've seen some of the photos, but I literally just took a picture of, from my own cell phone with the team in the lab,
Right.
one of the selamectin in a normal water solution and then a selamectin applied. And
Yeah, I've
lots of
seen
work
that.
to
Yeah.
still do, lots of tests to do, but very exciting, validation, that first step. And perhaps the best way to describe this quest for solubilization is just, the more soluble a drug is, the more effective its ability to be delivered into the body.
Right.
And without kind of getting too technical, it's like if you think about things that are not soluble, of course, your body's going to have a harder time, either getting it to the tissues or where it needs to, for the medication. So the more soluble it is, the more precision you have in that
Yeah,
case.
so I have a question about that as well, actually. So basically,
Mm.
when it's through IV, right, is that already 100% bioavailable? And then it's just like certain drugs like ivermectin. I know you couldn't before have it in sort of an IV form. You just had it in an oral form, right? So you guys are working
Yeah.
on, like, for ivermectin, for example. Now you have like higher solubility because of the oral format, and then you're also working on a kind of a needleless injection there as well, where it's basically just goes through your skin, right? Or
Yeah, so let me give you the broad. So ivermectin is a great example, especially to the IV question and where we see
yeah.
this all coming to life. So again, ivermectin is another one in that class of drugs. What's unique about today, there are basically harmful organic solvents that are added to make it
Right.
more ethanol and these things that wouldn't make it safe for human injection. So just to be clear, they're even in the husbandry animal category and such. There's what I've described more of a molasses-like texture of ivermectin. So our technique, makes it essentially 5,000 times more soluble. It's now the viscosity of water. And then to your point, well, what do we do with that? Where can we apply a solubilized ivermectin? So the first format we did was a rapid dissolve sublingual wafer,
Yeah.
which is essentially something the size of a piece of gum, chiclet, if you will, that just dissolves under your tongue. And then, of course, we have the fully injectable liquid format. And so, how
Which has
those
never been
apply
done
that?
before, right?
Correct.
Yeah.
So again, not without the use of organic
Right.
solvents that are harmful. So now we use two of our main excipients are already approved by the FDA. So I've gone on record saying we believe it's the leading candidate for any injectable form because it has no other harmful excipients. And if you start to imagine, even in our preclinical data, I describe why is solubility the holy grail. Well, it's just this candid. So, in the oral to oral form, in the existing oral form of ivermectin to our sublingual form that was made from the solubilized format, we essentially achieve the Cmax or the peak, drug levels within an hour versus 6 hours. So that's a pretty significant thing. Variability drops from 40% to 5%. And what people don't realize, as you take drugs into your body, you're essentially overdosing even on an aspirin, for example, because your stomach enzymes and liver and kidney, everything that filters, designed to protect your body. So the ability to deliver something sublingual is why you get the less variability, because it's not going through the stomachs and such.
And so essentially when you say variability, you mean like instead of when you take a drug, it spikes and then sort of levels out. It's like kind of at a solid level the whole time, right?
Yeah. Well, and variability impacts like your body weight. Did you eat? What's in your, digestive systems? Right. And, even if you've taken a pill before and you have that sort of feeling in your stomach, and all of those are contributors to a drug's variability, meaning how effective will it be? So they sort of take their best guess based on your weight, and there'll be 1 or 2 tablets, those kind of normal prescriptions. Well, the more variability, the less control, the less precision, the less variability, the more control, which is sort of the final point. Our preclinicals demonstrated that we're able to use 1/5 of the drug at a 5% variability for a faster onset. So even using less of a drug is one of our objectives, the less that needs to go into your body versus that, I use air quotes because I don't want to give overdose the wrong, term, but you're essentially overdosing on other applications where if we could do less more precisely. I should answer though clearly your injectable one, because just to kind of go back to the IV application. So that's the oral to oral. Yeah. Now taking the exact same, the solubilized format that is now injectable. What we're able to do in the preclinicals is take a subcutaneous injection, which is done currently with a 12 or 14 gauge needle, which is a thick straw, if you will, for your audience. And, typically, those are done in heavy animals like swine or cattle. They're not done in prize horses, for example, because of the scarring that's left. Those animals need to be, restrained because of the pain. And now you take something that's the viscosity of water. You can go down to a tiny insulin type needle, if you think of the comparison. And what we're able to do in our test is we were able to demonstrate a 0% variability with a 15-minute onset versus a 36-hour subcutaneous. That starts to change everything when you start to imagine that. And then now fast forward directly to your question on, how would this be used in IV drips or frontline COVID therapy as an example? The ability to inject someone at the hospital level or put them on an IV drip prior to maybe putting them on a ventilator, it would open up, much faster and more precise opportunities to treat someone that, again, might be on the front line with COVID that they've had to deal with.
Right. So going back to kind of that oral application where you're saying there's less variability as well, could that be applied to like an ibuprofen, for example, too, or?
Absolutely. Like what's really cool about our master desiccated liposome patent is essentially we take any molecule and we work towards formulating it to use less of it and apply it more precisely. And so again, we've done stuff in nutraceutical space with mushrooms. We've taken a cordyceps extract, which is I don't know if basically it's a mushroom that oxygenates your body. So it's very high in the energy space. And there we've demonstrated the ability to use 1/50th of the application versus the tincture.
Right. Yeah. I saw that. Yeah. Congratulations.
Yeah.
On those first revenues too.
Yeah. Yeah. Very cool. So thank you. Yeah. And so if you now imagine it's just what molecule, who wants to fund it, what's their business case?
Right.
Obviously the more expensive the molecule, the more fragile, the more value that some people get from it. But ivermectin is a great example too, where that's a generic drug. It's a very, very cheap, inexpensive, highly effective, drug that just, when you start applying it across these applications, we believe renewing, a 20-year patent in the generic space could be extremely profitable.
Yeah, I saw that. That's super interesting too. That basically you can kind of repatent and patent other ideas using your technology again, sort of for these. And usually it's only a 3-year patent, is what I've heard. So how does that actually work? Yeah, because that's really interesting, I think.
The 3-year you're referencing is likely if you think of the normal pharmaceutical development pathways. So they'll do initial formulation work. They think they're onto something, they'll file a patent. And then, as you know, it could be 10, 15, 17 years by the time they go through all the phases, all the trials. And often they're left with, a short competitive window where they can go to market and have a competitive,
because they've already had the patent for those 17 years before.
The patent protects you out of the gate, but you still have to bring it to market. Right. The benefit of us using drugs and vaccines that already exist, we're not a drug maker. We're actually applying delivery technologies to that. So of course, our patent window is much longer because we're not wasting, 17 of the 20 years on, trials in phase 1, phase 2, phase 3, all the things you need to do for the safety.
And that's because it's already drugs that are on the market.
Yeah, exactly. Right. We're already taking, things that are already there. Yeah. Mm-hmm. Correct.
Okay, cool. Yeah, that kind of brings me to the polio vaccine kind of that you guys are working on or trying to make more sort of effective there as well. So, that kind of goes into for the polio vaccine. I know for the OPV, right, you would in that case you would be making it more dose sparing with the bioavailability as well, and kind of that aluminum adjuvant as well. I know you guys have, but is it mostly for sort of cold chain issues as well? Because I know, because that kind of goes back to there's the IPV as well, right, of the polio vaccine. So the IPV would already have kind of that 100% bioavailability, right? So is this more than for the OPV case and the sort of cold chain idea, or then does the IPV also become more dose sparing with that kind of aluminum adjuvant and stuff?
Yeah, so let me just sort of jump. You're introducing a couple things that maybe make the question more complex, just given the different technologies. Yeah. But maybe if I just simply state, what is it that we're doing with IPV? We're actually not doing anything with OPV, but really? The IPV work that we're doing will, in fact, eliminate the need for OPV is where we're headed. Right. So essentially, we, last summer, we did our first work laying the IPV, the polio vaccine, essentially in a rapid dissolve format. One of the main things is, can we protect, the molecule, desiccate it, lay it down into our format? And that was where we started with the FDA's polio lab. And that work was amazing. It was a big proxy for, different pathways that we were working on. Then we started down a path now, which is, I think you're rightfully reading about and bringing up with cold chain and a dose-sparing adjuvant. We've stayed with this polio vaccine method because it's just something that we have a lot of people interested in, given the work. For example, Dr. Qsome is the gentleman who heads up the FDA polio lab. His father was the inventor of the OPV. And so there's a lot of legacy and, things that are important to the work that we're doing there. And so we've been pursuing that in a just basically, help supporting that cause. And then you may have heard about our principal inventor, Mike Farber. His father also did have polio. So this was just a project that is really cool. So a couple things to note. One thing is there's 7 categories of vaccines that, on a fragile scale. There's a single, which happens to be OPV in category 1. There's another single drug in category 2. And then the IPV we've been working is it within a list of 3. So it's the third most fragile and heat sensitive, if you will, vaccine. And that's important because the work we're doing is a proxy for other vaccines, even though we're focused in, as I just said, on the polio side. So what's really cool about this project now, to sort of link your question back to how does cold chain work? How does this work with the dose-sparing adjuvant? One of the things in our conversations with a pharmaceutical partner who's very interested in this work, I can't really go into a lot of the detail about those conversations under NDA, but I can tell you they were very instrumental in this kind of comes back to the variability and the precision that we talked about earlier. As much as there's a rapid dissolve oral application for certain vaccines, the hypothesis that they had was that they would prefer to still have an injectable format at the end user site. So there's an administration, there's a precision, there's a trained infrastructure globally, and particularly in third world countries, that already know how to inject. There's a lot of systems in place versus introducing any variability through an oral application. So that actually was a good learning for me early on, because there's certain applications maybe we can talk about in a minute around like insulin or some other categories where needle free is a really big target and a big desire. In this particular case, not so. So now where are we at with that? So interestingly enough, the cold chain work we're doing now is laying our, again, knowing what I just said about the desire to have a needle injection at the other end of it. So what will we do? So now we're laying inside a vial instead of producing a rapid dissolve sublingual product. We're using that desiccated liposome, laying it inside a vial. That project, I can give you the breaking scoop here today, is probably about 30 days behind schedule. In fact, the team was in just on the weekend to finish off stuff. We basically had a couple of our main pieces that were just delayed from COVID, so waiting on suppliers. But that literally ships today to the FDA polio lab. So that cold chain work that we've been working on, so that was pretty cool. The team again hustled over the weekend, and we're laying that again in a vial. So that trial is scheduled to test at 5-degree increments up to 40 degrees Celsius.
Right.
And yeah, what's amazing about people's understanding of cold chain, we didn't even hear it a whole lot until the recent vaccine stuff, because cold chain's not a common thing, but cold chain, simply put, is drugs and vaccines are produced in this 2 to 8 degree bandwidth, and they get shipped and moved around the world inside of that, temperature band. And then there's a ECTC and a CTC, which are just other bands that say, hey, how long can something come out of that temperature window before they in fact need to be disposed or spoiled. And so the significance of producing something in cold chain or that would break cold chain is significant when you start to imagine the logistical cost. Like, what does it cost to ship? And I've read reports over 17 billion a year is spent on cold chain logistics. And then in the vaccine space, another 35 billion of spoiled, wasted vaccines that can't be administered. And in third world countries, they don't have the electricity infrastructure, right? So you have to, work hard to navigate that entire structure. The other big part is, when you start to go down cold chain relative to our work on the dose-sparing adjuvant, where will those projects intersect? So the first thing I would say is the dose-sparing adjuvant is completely separate from cold chain, although we will bring them together at the end. So the dose-sparing adjuvant work, that's a trial that's going on right now with Tulane. And we've been able to take basically we're forecasting 1/10 to 1/20th of the IPV, also working with the polio vaccine and going through a process that again, would be applicable to other vaccines. And then there will be a second project that brings the two technologies together. Although each of them on their own are massive and significant,
Right.
we for sure are where you had asked in your question, thinking about the power of those technologies together, using a fraction, printing it inside a vial, shipping it outside of cold chain, rehydrating with distilled water in a local market, and injecting with the precision that pharma values.
Right. So you're still using an injection after. So you basically like
Yeah,
make
you
that,
rehydrate. Yeah.
right, rehydrate it, and then inject it like a regular IV would.
Yeah.
Okay.
Exactly. And that gets back to the 0% variability, the precision, all of those things we talked about.
Right. Very interesting. Yeah. So for the polio vaccine and the IPV there, would it be still, better than sort of the IV that's available now? Like, would it still be more soluble? Would you need to use less still than currently, kind of? Do you know what I mean?
Yeah. So when the two projects combine, for sure there'll be that
Right,
dose
when it's
sparing
together
implication.
because of the
But just remember, on its own, the vaccine on its own already delivers incredible things if it can, in fact, ship outside of cold chain.
right.
So
Yeah, no, that's obviously the biggest issue, is the logistics behind it with the third
yeah,
world countries, for sure. Yeah,
yeah.
no.
The desiccated liposome for sure always offers a bit of dose sparing. You don't get to the 1/20th kind of magic of a dose sparing adjuvant, but that's why combining those two projects in a second phase would be pretty impressive.
Right. So that's kind of where other vaccines would take place. You wouldn't be looking to basically use your Qsome technology and take a vaccine to take orally. It's more to kind of that dose sparing and that, right? Like say for the COVID
Yeah,
vaccine,
so
like say you take that, you would be really trying to make it more dose sparing, make it so it could be used outside the cold chain. But
the way I
saying
would
rapid
say,
dissolve is different.
I think it's important to understand. So oral applications absolutely are possible. We've already
Yeah.
that was the original work with the FDA, the ability to lay something down into a rapid dissolve format.
Yeah.
So again, just on polio, this particular discussion we've been having in a business development lane was the opposite of that. They don't value a rapid dissolve oral strip. So
Right.
let's just take a hypothetical, maybe something that's administered like the flu shot. That would be a good example, where imagine a nurse now overseeing a room full of people and everyone's taking a rapid dissolve oral format. That would be absolutely possible, where they could do a supervision of 20 people rather than lining up needles, injections, all of that stuff.
Right.
So that's there. And then also we're
that-
doing work
Or
in
sorry.
insulin for the same thing. Yeah, no, I was just going to say, in insulin, someone that gets a needle every single day or multiple times a day, those categories start to be pretty important, right? For just human, pain and suffering, if you will,
Right.
convenience, you're out at a restaurant. So the work we've done on insulin, for example, with Humulin R, you would take, 20 minutes, 30 minutes before your meal. Normally you're injecting yourself some predicting. Now you could take 5 minutes before. You could take a certain color, oral rapid dissolve format and start to offset the glucose spikes.
Right. So my question about that would be, would it taste bad? Like, that was a big question I kind of had. Like, would it taste like? Is it different for every drug, or?
Yeah. So the cool part of our desiccated liposome format is you can essentially mask the flavor profile of anything.
Right.
So we're doing advanced work in even something. Even it's funny, our sleep product has a melatonin and valerian root. Valerian root on its own, if I had a jar of it in this room, everyone would need a gas mask.
Right.
But you're able to take it, layer it, and complex it in a way that can mask its smell and flavor profiles. And so each molecule has different things that the team would work on. But yeah, so and then where you can legally add flavorants as well. So for example, we're doing work in the CBD, THC, cannabinoid space. They're not naturally great flavors, but the ability to mask it and layer it on. So in the edible category, there's a lot of regulations about flavorants and, you know, making things so it doesn't appeal like candy, if you will, for young children. So using natural, you know, mint and acai berries, or however that's pronounced, you know, you've got these certain natural flavorants that are deemed a preservant, you know, that would help, you know, mask the flavor profiles. So yeah, lots of cool stuff can be done on the flavor.
Yeah, that's really cool. So kind of, I know you just sort of mentioned the mushroom aspect there, what you're doing there. And so what I'm wondering, because of the license agreement there, you kind of have well, I know you got the original payment, and then you also have sort of a license agreement per sort of the sales of the product. And you also took some a percent of the company, like you got some shares in the company there of $50,000 worth. Are you gonna always kind of plan to take some equity stake as well in sort of some of these smaller companies that you'll be working with there?
Yeah, it's a great question. It's not a standard answer. It would depend on the deal framework. So on that particular first deal with Circadian, when we did a land transaction, we also took a fairly large, like almost 10% equity stake in them at that time. And then so this additional, and they're going through rounds of funding in that. The reason we're excited about the mushroom space, again, we're not looking to get into the mushroom space from a cultivation production, but to apply our technology with a partner in a massively exploding space, gives us a little bit of upside. It gives us a bigger return on that original land deal. They're already raising money at higher gates, so that's been a really, really good deal. But it's fair to say those deals are never certain, right? Companies can go bankrupt. They may never go public. There may be no exits. So we don't want to do those deals in every deal, but in the space that's right, and after doing our diligence with a partner like that, that one made a lot of sense for us.
Yeah, no, I think it's a great idea for sure in that case as well. So that's cool. Then going off that as well, so you essentially get royalties per kind of sale of whatever the company's selling, right? So is there like a general percentage you guys usually get from that, or is that something you can
so that's a good kind of broad question that I could start with on what is, you know, how do we bring molecules to market? Our original business plan has been to take a molecule, find a partner, license our technology, and then make a royalty stream, you know, with that partner. Each deal is different. Each molecule is different. Some, you know, even we're far along in a deal in the cannabis space as an example, and there's 3 or 4 layers to a deal like that. One is just a simple license technology. They manufacture, they sell it, and we secure a percentage of either the markup or percentage of retail, depending on what we're doing. In something where, you know, in cannabis, it's quite interesting because the molecule is very expensive. So, you know, there we have a higher leverage point because there's something that completely differentiates them with us, without us, is how I would describe that. So the second piece of that type of deal might be, hey, if Mountain Valley were to produce stuff for another partner and broker that deal, perhaps we also have them, you know, one company manufacturing for a larger, you know, white label versus their own products. And then a third might be, hey, we bring large deals in from a production standpoint that have nothing to do with us, but they came in. We might also take a royalty of that deals. You know, that would be an example of how we're working through, you know, different potential lanes. The Circadian deal in particular, I'm not going to disclose exactly, I think just in respect to their business model, but we're in that deal, we're linked to a percentage of retail. So essentially everything that they sell, we get a percentage back down on. And the money that they pay us They also pay for all the formulation work, lab work, sample work, as you would expect, just as a business model. We don't do that work for free either. So we would help a company, teach them to fish, license our technology, and then do all sorts of ongoing product formulation work in the background, and then take the royalty stream. And that satisfies, by the way, the scale to our business, which is a logical follow-on. Like as we work with partners who can produce and manufacture, that's a lot, easier margin, if you will, for a company like ours. We don't have to scale up all these different production lanes.
Right. Yeah. No, that's nice to hear. Kind of you're doing it per deal kind of basis.
Yeah.
That's awesome.
Yeah. The value of the molecule drives the common sense to it, candidly. For example, some of our early work in cannabis, as a better example, when you start to now, so today there's a 10 milligram capacity on an edible, certainly here in Canada. So we are able to wrap that into a liposome, in a desiccated format under a patent. If we can deliver something now that has a 4 or 5x in its impact and its potency, but still stay under the 10 milligram legal limit, or go to a 5 2 milligram pills in a 10 milligram pack, as an example, the value of that to the end consumer, as would be, I'd have to go and buy 4 or 5 of those edibles. Whereas now I could have one of these. A lot more margin to be made in that formula. And so we'd want something much larger than a typical nutraceutical deal.
Right.
But again, in a win-win, we want our partners to be
obviously
the most incented, and most profits go there. We would just want our reasonable cut that helps them be successful.
Cool. Yeah, that's cool. Now I'm wondering about the sort of FDA approval for the Avectisol that you guys have, I guess, coming up pretty soon here as well. You're going through the 505(b)(2), and that's essentially basically means that you need to basically, you can skip a lot of the steps of some of the some of what's already been proven, sort of FDA approved, right?
So they don't skip things, but they take things as a proxy of work that's already been done. So they still require the same discipline, of course, but they'll take a safety data as a proxy, for some of those gates. So you're correct in skip, but you don't get to hop over steps. You get to just, have other things in for that.
Sort of as precedent there. Okay.
Yeah.
That's cool.
And so that does give us a faster track for sure, which is, the one question you asked before that I realized I hadn't fully answered around needleless, and even in the injector systems and what we're doing in the husbandry animal category. This is a good example. So what is the 505(b)(2) pathway? That is a direct solubilized human application. And that one is incredible. We're really excited about it. In the meantime, we have husbandry trials that are going after needleless injectors. And why is that important? Well, you have now, first of all, globally 100 billion potential injections. When you look at just swine, cattle, and poultry, I think that's almost 70 billion alone. Taking a needleless injector, which essentially just opens one hair follicle on an animal and does a needle-free injection. So it's very precise. It eliminates a lot of handling issues with needles and safety issues, even breaking off needles into food supply. People don't realize how common that is. So it takes care of a lot of problems. There's a speed and efficacy, a precision, et cetera, et cetera. So we're very excited because earlier in the interview, we talked about the subcutaneous, the thick straw, you can inject poultry, for example, in that mink and some of these smaller animals. That would be a massive new opportunity for ivermectin. And when people sort of ask me, what do you get excited about? What's the big deal about ivermectin? It's not just because of COVID. That just happens to be exciting and relevant. But we originally went after this broad parasitic category for all sorts of applications across husbandry and companion animals. And that's how you get to mosquito-borne viruses and a whole bunch of business lanes that we're pretty excited about.
Right. Yeah. No, it's going back to kind of that thing you said about COVID, right? Because it's interesting because you're not really, what you're doing there is kind of taking the ivermectin and distributing it in like a group, like a smaller population group, right? To kind of mitigate sort of that whatever virus it might be, right? And you would do the same thing in the husbandry animals in this case, right? You would give it to like a certain population so it doesn't continue to spread. Is that
Yeah.
true?
So exactly. Like if that would be a good example of mosquito-borne viruses. So I have gone on record before describing an ecosystem, and maybe just for your mind, if you think a village or a small town, just because it's simplicity, you have now the ability to dose humans, every class of animal, companion like dogs, cats, etc., and then every farm animal, right down to poultry, small game, all of that. Now you can essentially create a reservoir that would kill anything a mosquito feeds on of ivermectin. It would be a poisonous dose to the mosquito, but a safe dose in the human and animal population. And so, malaria, is the one I always imagine. By the time we're done this interview, there'll be 15, 20 children that have died from malaria, as an example. And so there's a lot of amazing work, but this is the power to unlock something that can truly eradicate malaria and the scourge of mosquito-borne viruses, Zika and dengue, and there's a vast list of them.
Right. Wow. Yeah. No, that's awesome. I think you guys are doing amazing work there, and
Yeah.
yeah. No, I think you guys are going to do really well. I'm going to continue investing as well. So
Yeah, it's awesome. We're a great story. Again, we appreciate your support, helping us get our word out. There's no doubt, the stuff that we have coming even in the next 60 days and some of the trials and
yeah.
results will continue to generate a lot of excitement about our company.
Yeah. Cool. Well, I guess you're probably pretty close to out of time here as well. Hey, like.
Yeah, no, that's good. Well, hopefully that covers. We can always schedule a follow up if
Yeah, no, definitely.
you want to
I
go
would
a little
love to
deeper.
have you on the channel again. Talk to you anytime, really. Yeah, yeah. This is actually the first interview I'm doing on my channel as well. So
Oh, cool. Well, good for you. I hope it does well for you.
yeah, I'm sure it will. There's a lot of people out there who are really interested in what you guys are doing, and it is sort of a more difficult kind of space to understand. So
Mhm.
I think it's really good that you're doing like these interviews with smaller YouTubers and stuff too, and just kind of sort of helping people understand what you guys are doing and everything. So
Yeah, cool. Well, send me a link when you get it up and running, and we'll
I
help push it
will,
out in
yeah.
our network. I'm sure our users would value this discussion as well.
Yeah, no, definitely.
Awesome. All right. Thanks. Have a great day.